Welcome back viewers to this channel. So in this video we will be discussing about protein folding and denaturation as well as Anfinsen experiment of protein folding. Protein folding is a process in which this polypeptide will fold into its native three-dimensional conformation, and this process may occur before or may be after biosynthesis of protein. But there are also some factors on which this protein folding process may depend, and they are nature of solvent, concentration of salt, temperature, and molecular chaperone. So in some cases, when there is a high concentration of salt, high temperature, and absence of molecular chaperone, this protein may not able to fold into its native conformation, and protein will become functionally inactive. So protein folding process is necessary because protein will become functionally active only when it will attain its native conformation. Now when protein loses its native conformation, they become denatured protein, and the process by which this protein loses its native conformation is known as protein denaturation. It includes the breaking of non-covalent bond like electrostatic interaction, hydrogen bond, and hydrophobic interaction, as well as covalent bond like disulfide bonds. But this does not include the breaking of peptide bond of the protein structure. Now, since it only includes the breaking of non-covalent and covalent bond, protein denaturation includes the loss of quaternary structure, tertiary structure, and secondary structure of protein, and it does not include the loss of primary structure of the protein. Now, there are some reagents or conditions that can cause denaturation of the protein, and such reagents are known as denaturing agents. For example, strong acid or bases, when there is a change in pH, it will result in deprotonation or protonation of the side group, and it will disrupt the hydrogen bond and disulfide bond of the protein structure. Organic solvents like ethanol, it has the ability to form hydrogen bond with the protein structure, and it will disrupt the intramolecular hydrogen bond of the protein. Detergent are amphipathic, it can be ionic or non-ionic, and it disrupts the hydrophobic interaction of the protein structure. Reducing agents like beta mercaptoethanol, it has the ability to reduces the disulfide bond into sulfhydryl group, and it disrupts the inter as well as intramolecular disulfide bonds. Heavy metals like mercury and lead, it has several ways to disrupt protein structure, and one of it is that it can disrupt the salt bridges of protein by forming ionic bond with the negatively charged group. Heat, when it increases, it results in increase in vibration, and it will result in breaking of weak interaction like hydrogen bond and van der Waals forces. Now let us understand in better way about protein folding process by the experiment carried out by Anfinsen in the year early 1960s. For his experiment, he took a protein named ribonuclease A. Ribonuclease A is a pancreatic enzyme which has the ability to digest single-stranded RNA molecule. It has a molecular weight of 13,700 Dalton, 124 amino acid residue, and 4 disulfide bond. Now what he did is that he treated this native ribonuclease A with urea and beta mercaptoethanol. Urea is a denaturing agent and can break hydrogen bond of the protein structure, and beta mercaptoethanol, since it is a reducing agent, it breaks the disulfide bond of the protein structure. So when ribonuclease A is treated with beta mercaptoethanol and urea, the protein becomes denatured or reduced, and all its 4 disulfide bond has broken down. Now when this denatured protein is allowed to renatured again in the absence of urea and beta mercaptoethanol, he found that the protein has attained its native three-dimensional conformation. So his first finding is that all information to fold into its native three-dimensional conformation is present in the amino acid sequence of the polypeptide chain. So again, in his second experiment, what he did is that he allowed this denatured protein to renature in the absence of only beta-mercaptoethanol but in the presence of urea. So what he found is a scrambled species of protein, and this 4-disulfide bond has formed but in incorrect position. So his second finding is that weak interaction or hydrogen bond are necessary to form its native conformation. So this is all about Anfinsen experiment of protein folding.